Fantastic. Okay, thanks, Anna, for the introduction, for the invitation. I want to also thank Manuel for giving us the opportunity to be part of these seminars. It's very kind of you to consider our lab, our research, to be part of this amazing list of speakers and groups. As Anna mentioned, we opened the lab in Cincinnati Children's about a year ago, so everything is so new and we have many projects going on. One of the key questions we are trying to understand and investigate is mechanisms at cellular level of integration of internal and external signals. Particularly, we work on chronobiology and circadian rhythms, so we care about the way that circadian signals affect the communication of neurons. So if we think about language, these neurons are speaking to each other, having these phasic changes across the day-night cycle. But in addition, we experience the night-dark cycle. So there are external signals, different language that needs to be encoded in these circuits, controlling the function of these cells and eventually the function of the target of these circuits. So we have a very interesting scenario where a neuron is now receiving information from homeostatic signals from other brain areas, the body, different organs, but in addition, information from the environment. We're part of the ophthalmology department, developmental biology. We care about light in particular, because we know that light has a drastic impact on physiology, and in particular because of the drastic consequences of irregular light exposure. We are all exposed to different irregular light conditions, but in some way we can control it. I hope so. So we have good opportunities to expand our working hours and do whatever we need with light, but there are people that don't have so many options. And if we think about shift workers, for example, these people are exposed to different LiDAR cycles that can be changing every week. And it has been extensively reported that for long exposure under these irregular light conditions, there are many symptoms that can be identified. Some of them listed here, not only circadian and sleep alterations, but also deficits in metabolism, cognitive performance, and mood or affective behaviors. So one of the key questions in the lab is understanding the retinal brain circuits and mechanisms that drive light-mediated deficits. In mammals, the retina is the tissue is part of the nervous system that detects light and routes this information to the brain. In this beautiful schematic from Santiago Ramón y Cajal, you can see the different layers of the retina where you have classic photoreceptors, rods and cones that detect light. This light detection leads to processing of this information that the photic input is now transformed into electrical and chemical signals that through these interneurons in the retina, the signal reaches the ganglion cells, the projection neurons of the retina, and therefore all the information is routed to the brain. We are visual animals. We consider have a strong connection between light and vision, and this is mediated by the projection of the retinal ganglion cells to visual centers in the brain. Two examples are the dorsal geniculate nucleus, the part of the lateral geniculate nucleus, that is the first station processing light information for vision. The DLGN is then connected with the cortex, the visual cortex, controlling image forming functions. In addition, there are projections to the superior colliculus, an area that is involved in integrating multisensory information. For example, when we hear a sound, we move our head trying to identify the source of the sound. This integration between hearing, vision is coordinated in part by the superior colliculus. So we can talk, at least in humans, about conscious vision as a process controlled by the retina and these visual centers. But in addition, we know that light is important for regulating our internal rhythms, our sleep patterns. About 20 years ago, or even more than 20 years, it was characterized and identified a cluster of ganglion cells in the mammalian retina that express a photopigment called melanopsin. So these neurons are similar to the conventional ganglion cells. They have projections to the brain. They have similar dendritic arborizations, but they also express the photopigment melanopsin. So these are atypical photoreceptors that were originally described as intrinsically photosensitive retinal ganglion cells. We refer to them as iPRGCs. It's very interesting that since then there's been a lot of studies and research on these neurons. And we know that in addition to this intrinsic photoreception, the iPRGCs can directly sense information and receive information from rods and cones. So we have a first step of integration of internal and external signals. Okay, these neurons are processing light input from rods and cones, similar to conventional ganglion cells, but they also blend this information with the photoreception processes, with the detection of light through the expression of melanopsin. These neurons are depicted here in red with immunostaining for melanopsin. They are about 5% of total ganglion cells, but even though they are a small percentage of neurons, they have a dense innervation to the brain. And one of the main areas that was originally characterized is the suprachiasmatic nucleus. So this is a target of iPRGCs that in David Pearson's paper, 2002, was the basis of all these studies where they found that projections to the SCN, these neurons are responding directly to light. So they are intrinsically photosensitive retinal ganglion cells. So when we talked about circadian rhythms and light, we're actually talking about photoentrainment. And this is the process of the circadian system, these brain centers processing external information to control their internal rhythms. And one of the easiest ways to control and study this is by looking at the locomotor activity. In the lab, as many labs in chronobiology, we are using mice as our animal model. First of all, because we can do a lot of genetic manipulations. And in addition, these are mammals with similar anatomical features compared with humans. So we can do a really good analysis and extrapolate results to human research. What you can see here on the right panel is an actogram. We refer to actogram to this plot where we can see every day what the animal is doing. So here from 0 to 24 is day 1, 24, 48, day 2, and then day 2 is repeated here. So it's a visual way to understand what the animal is doing. In this case, it's an animal on wheels. So we measure the wheel activity or revolutions on the wheel, but you can also use infrared sensors, feeders, many variables that give you information about their rhythmicity. These are nocturnal animals, and you can see this here. In normal LiDAR cycle, light is represented in white, gray is the dark, the night portion of the day. These animals are mostly nocturnal, but we can then play with the lighting conditions and mimic, for example, shift work or traveling across time zones. So here is an example of a person that travels from Europe to Argentina, Argentina to Europe, India, anywhere with different time zones. And as you can see, they need a couple of days to realign with the onset of the dark portion of the day. So we can quantify this, have an idea of how they respond to the environment. We can then delay the onset of the night portion of the day and measure how long it takes again to re-entrain. But the most important aspect of all this is when we housed animals in constant darkness, you can see that the internal rhythmicity is preserved, indicating that the SCN, the suprachiasmatic nucleus, and the circadian system as a whole is still rhythmic. And these animals can now predict or anticipate the beginning of the next day, or what we called the subjective day or subjective night. Okay, so this is very precise. You can see the free running period of animals and even small light bulbs. It's shifting the activity a couple of hours, but then again, they have a clear free running activity. So internally, the system is controlling the activity of the animal without any external cues. But at the moment you present light bulbs, these neurons are integrating these internal signals with external signals to control a coherent output behavior. At the molecular level, we have now a really good understanding of the clock genes that regulate these responses in neurons and cells in the organism. This is a very schematic and very simple representation of the clock. I invite you to read more in many of Takahashi's reviews and some other good researchers doing amazing job on this, where we have a good understanding of the clock genes involved in these rhythms. As shown here, it's an example where pair and cry clock chains have evox regions on the regulatory region that are controlled by the complex clock and VMAL. So when this complex is binds to the evox, there is expression, and you have the proteins pair and cry in the cytoplasm of the cell. Eventually, with time, there is accumulation of this complex. They can translocate into the nucleus, leading to the they can now block clock and VMAL having an autoregulatory signal. So it's a very precise mechanism that it's cycling every 24 hours. Okay, so in this plot, I'm trying to represent the different feedback loops of this mechanism have different peaks of expression, but in all cases, the rhythms are close to 24 hours. This is why we talked about circadian rhythms. Circadian means circa DM, close to a day. So we have clock gene expression in the SCN, but also in many organs represented in this schematic. Virtually every cell in the organism have a clock that is controlled or orchestrated by the central pacemaker in the SCN. So this leads to the central dogma in chronobiology, where central clock controls the function of peripheral clocks. These peripheral clocks can be located in the body, as shown here, but also in many brain areas. We have now really good understanding, and there is a lot of research expanding our understanding of clock gene expression in the brain. There are many areas that are considered peripheral clocks within the brain, and even the retina can be considered a peripheral clock within the central nervous system. As shown here, LiDAR cycles are the strongest time cue in nature. They direct connections with the SCN through the retinal projections, as I mentioned before. And these external signals synchronize rhythms in the SCN, and therefore signals, humoral and neuronal signals, control the function of peripheral clocks. So this was the central dogma in chronobiology. It's working great for all of us, but it's a little bit more complicated than this. We have now a better understanding of the impact of external cues on peripheral clocks. So in addition to the central pacemaker, many cues in the environment, in addition to light, food, temperature, social cues, stress, they can directly influence the function of some of these peripheral clocks. So it's a more complex scenario where we can investigate, for example, how light affects the SCN, but also how directly impact the function of other brain centers. And this is supported by this new schematic of IPRGC projections to the brain. In 2002, these neurons were characterized and the connections with the SCN were described. And since then, we and others have been doing extensive analysis on the pattern of connectivity of these IPRGCs. And this is what we now know. It's possibly not complete because there are, again, many publications, but if you have an understanding of brain anatomy, you can see how diverse the targets are, and clearly they go beyond the SCN. You have areas in the thalamus involved in vision and non-visual functions, limbic centers, the lateral habenula, superior colliculus, as we were talking before, as well as many areas in the hypothalamus. So all this characterization started with the use of good genetic tools, the generation of animals with the expression of the Cre recombinase in melanopsin positive neurons. In this paper, this is a really nice example where researchers use animals where the melanopsin, sorry. The melanopsin neurons are expressing the Cre recombinase, and this recombinase can now induce the expression of the genes that we want using adeno-associated virus that can be injected into the eye of the animals. So in this example, they injected an adeno-associated virus. It's a Cre-dependent virus expressing the reporter alkaline phosphatase. By injecting the animals, you can see in the retina all the neurons, all these melanopsin-positive neurons labeled in black, and then they can trace the innervation to the brain. As beautifully shown here, they confirm the projections outside the SCN. Obviously, the SCN is densely innervated, but in addition, you can see fibers reaching areas of the lateral hypothalamus, the supraoptic area, as well as dense innervation to the geniculate complex, okay? The DLGN, again, this is an area directly involved in visual functions. So we can now speculate of the role of these melanopsin neurons in controlling visual functions. In a similar note, we published a paper where we use a more sophisticated method for controlling the Cre expression, and this is the Cre-inducible system, also controlled by the melanopsin promoter. As represented here, the Cre recombinase, the inducible version, now requires tamoxifen to be in the system. It's a drug that we can externally add to the system by injecting the drug into the animals. And in the presence of tamoxifen, the complex can now translocate into the nucleus, causing the recombination and expression of the genes that we want. So by using this strategy, generating these animals and targeting small volumes of tamoxifen into the animals, we were able to generate protocols where we can induce the expression of the alkaline phosphatase in a single neuron in the retina. This is shown here. This is one of the examples that we have where you can see, one second, yes. You can see the neurons here, the dendritic arborization, so it's quite easy for us to identify the subtype of iPRGCs based on the anatomy, but more importantly, we can trace the projection of the axons of these cells, okay? In this case, you can see the axon going to the optic disc, and then by doing serial analysis of the brain, we can understand more about the innervation of a single neuron in the retina and how these neurons modulate the function of different brain areas. In this video, you can see the reconstruction. Again, this is a single neuron projecting to the SCN, mostly the caudal part of the SCN. And we found with more than 30 neurons that each neuron has a different pattern of connection with the brain. In blue, green, and red, you can see 3 different examples, random examples, that cover most of the SCN. And in fact, it was very interesting to note that neurons in the dorsal part of the retina innervate mostly to the dorsal part of the SCN. The same happens with the mid part of the retina and the lower portion of the retina. So this is suggesting a potential retinotopic map where the retina is driving information to different areas of the SCN and possibly giving information about time. If we think about rodents, they can see sunset sunlight. Depending on the angle of the light, different retinal cells will be activated, and therefore different areas in the SCN are being activated because of the iPRGC function. So this is a hypothesis. We are currently working on better tools to characterize the connectivity of these single iPRGCs. What is important is that in addition to SCN innervation, we found that most of the neurons projecting to the SCN, they have collateral fibers innervating areas of the ventrolateral or ventromedial hypothalamus. In this case, also the geniculate, the ventral part, the IGL, and superior colliculus. As we were talking before, if you have some understanding of brain anatomy in mammals, you can start speculating how complex these connections are. So we have in a scenario again where one neuron detecting light by the melanopsin expression can now modulate the function of many areas in the hypothalamus, the thalamus, as well as superior colliculus, controlling diverse functions associated with changes in light. And this is represented here, and is one of the conceptual frameworks that we are investigating in the lab. And I've been working also I was working when I was with Samer Hattar at NIH, the Mental Health Institute. The idea here is that in addition to the SCN, we have now a good understanding of iPRGC connections to different brain areas. So we know a lot about these brain regions, so we can speculate about the direct effect of light through iPRGCs over these functions and how these light signals can directly affect the function of these centers. And importantly, when we think about the symptoms associated with irregular light exposure, we can have the first step in understanding how light can directly affect the function of many brain areas that are directly controlling symptoms associated with irregular light exposure. So what do we know about iPRGCs? I wanted to briefly summarize 2 studies that I think were essential in linking iPRGCs, light, and innate behaviors. In this example, again, the authors use adeno-associated virus, OPM4 Cre mouse lines, but in this case, they injected animals with a virus expressing the red receptor. So the chemogenetic control of neurons is an amazing technology, an amazing tool that we've been using for many years. And it uses the expression of designed receptors that are specifically activated by drugs, in this case, CNO, that we can add to the system. So in normal conditions, CNO is expected to not have an effect on the animals, the same with the DRET receptor, but only when we inject the CNO or we give CNO to the animals, the receptor is now responding to CNO, and in this case, causing the depolarization of the cells, okay? Depolarizing the cell causes the activation of the neurons, so mimicking the activity of the neuron, in this case, mimicking the effect of a light pulse. And what they found is that this acute activation of iPRGCs, and importantly in animals housed in constant darkness, cause activation of many limbic areas, changes in locomotor activity, possibly through SCN activation, but importantly, they noticed increased alertness and anxiety. So these are responses directly controlled by limbic centers. We have candidates in mind that could be involved, but in any case, it's showing that acute activation of iPRGCs, a single dose of CNO, has drastic effect on many behaviors, in addition to locomotor activity behaviors associated with mood, anxiety, and cognitive functions. A few years later, a paper from the Hattal lab was, I think, a key result showing the role of iPRGCs in driving irregular light effects on behavior. In this case, Taraligate housed animals in an ultradian lighter cycle, defined as the T7 cycle. It presents 3.5 hours of light and dark, so it's 7 hours total. This ultradian lighter cycle for 2 weeks causes severe learning deficits and also alterations in mood-related behaviors without causing circadian disruption. So this is a great model to dissociate circadian disruption to learning and mood deficits in animals. Then she housed animals under the same condition, but in this case, these animals have the expression of diphtheria toxin in the melanopsin positive neurons. It's a genetic strategy to ablate iPRGCs, okay? So these animals have normal retinas. The development of the retina and the brain connection is completely similar to control mice, but in this case, the expression of the diphtheria toxin causes the ablation of the cells. So in adult animals, you can check and confirm the lack of iPRGCs in the retina, and of course, the lack of connection of these cells with the brain. So the SCN and many other targets, as highlighted in this model, are not receiving information from iPRGCs. Their vision is completely normal. The responses to light in some way are normal, but when they are exposed to the T7 cycle, they showed no deficits in learning function and also no deficits in mood-related behaviors, suggesting that iPRGCs are the main retinal channel processing irregular light information to affect these complex behaviors in mice. So at this point, we wanted to better understand the functions and the role of many of the candidates that receive innervation from iPRGCs. And we focused on an area of the dorsal thalamus that is known as the perihavenular nucleus. It's part of the dorsal thalamus. It's very close to the lateral havenula. This is why we called it perihavenular region, but it's exclusively thalamic neurons that are densely innervated by the retina. In green, you can see here the innervation of all ganglion cells, as expected, dense innervation in the lateral geniculate center, innervation also to the pretectum area. But importantly, we notice fibers that densely innervate this region of the dorsal thalamus, the PHB, as shown in this coronal section. With more tracing analysis, we confirmed that these are exclusively iPRGCs innervating PHB neurons. And what we also notice is that these neurons display rhythmic clock gene expression. As shown in these images, this is immunostaining. We are looking at the protein levels of PER2, the clock gene that I mentioned before in the introduction. So there is a rhythmic expression. We can see rhythmicity across 24 hours. But interestingly, when we housed these control mice under irregular light conditions for 2 weeks, similar to what Taraligate did before, we noticed that these rhythms are gone. And you can see the analysis and the quantification here. In red, you can see that there are no rhythmicity. There is no significant differences between time points. And importantly, there is a upregulation of PER2. As I mentioned in the intro, PER2 is part of the negative feedback loop of the clock machinery. So we can start speculating that the clock function is significantly disrupted, and therefore all the genes that are controlled by these transcription factors will be also affected. What about the projections? Well, we did a couple of tracing analyses to confirm that iPRGC innervated neurons in the PHB project to limbic areas as shown here. There are innervations to the nucleus accumbens, dorsal striatum, and the prefrontal cortex. In particular, we saw dense innervation in the infralimbic area. As you may know, these limbic areas are directly associated with mood control, with cognitive processes as well. And importantly, they are main targets involved in the pathophysiology of clinical depression in patients. So we have an interesting scenario where now in two synapses, we have neurons in the retina detecting light, projecting areas of the thalamus that directly control the function of the limbic centers associated with mood control. So we wanted to play with these circuits and use different strategies to manipulate the cells. And as shown here, we started by using the DREAD technology that I previously described. We use retroviruses to label PHB neurons projecting to the prefrontal cortex. And again, using a Cre recombinase system and Cre-dependent viruses, we induce the expression of the DREAD receptor. This is the excitatory version of the DREAD, exclusively in PHB neurons projecting to the ventromedial prefrontal cortex. We then housed mice under regular LiDAR cycle for 2 weeks, but in this case, we gave animals CNO in their drinking water. So we wanted to generate a normal LiDAR cycle where animals, as nocturnal animals, are drinking water mostly at night. So this is causing an irregular activation of the PHB mediated by the CNO in the drinking water. 2 weeks under these conditions were sufficient to significantly affect the behavior of these animals as shown here. You can see the force swim test, sucrose preference test, how there is an increase in mobility time for the force swim test, signs of helplessness, as well as lack of preference for sucrose that in some cases is associated with anhedonia or this lack for pleasure. They definitely are drinking water, so the amount of water consumed is the same, but they don't have a clear preference for sucrose as controls do. In addition, we wanted to silence the PHB, so do the opposite experiment where we can now investigate the consequences of animals exposed to irregular LiDAR cycle where only the PHB is now silenced. We were able to do this by injecting viruses expressing the tetanus toxin. Again, adeno-associated virus injected in the PHB. In green, you can see the injection in the PHB and some neighboring areas. But in all cases, the SCN is not affected. Many of the limbic centers, the hippocampus, none of these centers are affected. When we housed animals under the T7, control mice showed the expected deficits in behavior, but now animals with the tetanus toxin virus in the PHB, so neurons that cannot route information to any of their limbic centers. In this scenario, the irregular LiDAR cycle has no effect on their behavior. They are completely similar to animals housed in LD cycle. Combined, this data indicates that the PHB was sufficient and necessary for driving the irregular light effects on mood. And also in the paper, we characterized the projections to the SCN that are affecting the central pacemaker. And we have the hypothesis that is involved in affecting the hippocampal functions, leading to cognitive deficits. This is, again, under investigation. We need to do much more studies to better understand this arrow here. Regarding the PHB, this arrow is quite understood, at least the basics of it. We can simplify the model by saying that iPRGCs projecting to the PHB are the first step in modulating mood control centers, including the ventromedial prefrontal cortex. So when we started with this project, it was a few years ago, and I had the impression that it's been a revolution in our understanding of the thalamus. If you go to most of the textbooks, you will read that the thalamus is a relay station that directs sensory information to different cortices. So this is clearly based on the anatomical projection based on the fact that the thalamus is mostly excitatory neurons that project to the cortex. But we wanted to better understand this connectivity, and particularly based on many studies that were published in the last 5-10 years, investigating the local processing of sensory information within thalamic centers. So what we did is just characterize the anatomy of the PHB. As a new center, we had no idea about the cells. We expected to find excitatory neurons, and we found them. In blue, you can see RNA scope. Now we're looking at RNA expression, this topographic analysis of coronal section of the mouse brain. So excitatory neurons are everywhere in the thalamus, confirming what we read for years in papers. But also we notice a cluster of GABA neurons expressing BGAT in the PHB. These are approximately 25% of the total cells in the PHB. And we found that most of the GABAergic markers are expressed in these neurons. They have GAT1, GAT2, VGAT, as I showed you. And as you can see in this image, in the bottom part, they are clustered within the PHB circuit. So it was a very interesting result that was confirmed using reporter lines. In this case, we use a GAT2 cherry animal, where we do a serial sectioning of the PHB, confirming that these neurons are exclusively clustered in PHB. And as you can see, outside PHB, there is virtually none GABAergic cells in the thalamus. You can see now an analysis from rostral to caudal thalamic centers. And what we notice is the higher the retinal innervation, the higher the number of GABA cells, suggesting a close interaction between these two neurons. And in fact, we confirm this using electrophysiology and more tracing analysis with the rabies or monosynaptic tracing. And all the data confirmed that iPRGCs, in addition to innervate the excitatory neurons that are connected with limbic centers, they have projections to GABA cells within the PHB that locally modulate the function of these excitatory neurons. So we have regulatory system within the thalamus that can in some way control the flow of information from the environment to limbic centers or cortical areas. Interestingly, when we measure at the light activation of these cells in the PHB in animals housed under T24 or T7, we notice that after the exposure to irregular LiDAR cycles, the total number of cells that are responding to light is similar to control mice. However, the fraction of GABAergic cell that is responding is significantly higher, suggesting that these cells play a key role in modulating responses, particularly when light signals are abnormal. We are currently investigating this at the molecular level. We wanted to know more about what it means. Is it a break? Is it a way to control too much information? Is it a mechanism to control or preserve the function of excitatory neurons? We don't know the answers. We are currently working on this. But what we know is that we can measure bulk sequencing to know or to have a comprehensive understanding of clock gene expression in the PHB. There is, again, in the last, I would say, 5 years, a lot of papers now investigating clock gene expression in thalamic areas. There's great understanding of the expression in the visual geniculate targets. So we wanted to do a similar analysis for the PHB. What you can see here is a volcano plot comparing the expression of all the genes that are being expressed in the PHB of animals where the tissue was collected during the active phase or the inactive phase of animals, okay? This is comparable to day and night. Basically, we housed animals in normal conditions. We dissected the brain, and particularly the PHB tissue, to run RNA sequencing to have a complete analysis of all the genes that are being expressed. In red, you can see here all the genes that are upregulated during the active phase. Blue are genes downregulated during the inactive phase. If we cluster them together, we can see categories that are upregulated at CT14, circadian rhythms, regulation of gene expression, as well as cell-cell communication. During gene cell enrichment, we confirmed the role of GABAergic pathways controlling the function of the PHB, particularly during the active phase of the animals. The same for homeostatic processes during light phase. So this was in some way expected. The thalamus is rhythmic, controlled by the TRN, zona incerta. We know that phasic activity control the function of these thalamic communication with the cortical centers, but we wanted to know more about the PHB in animals housed under T7 cycle. So we did a similar experiment, but in this case, we collected tissue at the active phase, inactive phase of the animal, focusing on the locomotor activity, okay? As I mentioned before, this is a very interesting light paradigm because it's not causing drastic significant or drastic circadian dysregulation. As you can see in this model, every day we can anticipate the onset of activity. They are in something similar to a free running with periods close to 25 hours. So using the activity, the actograms, we can precisely measure the onset of activity, collect tissue at CT14 as well as CT2. When we compare the data, we're honestly very surprised to see the lack of rhythmicity. So this volcano plot, again, is comparing the expression of clock genes at CT14 versus CT2, okay? This is not reflecting the levels of expression. This is reflecting changes among these two time points. And what we notice is that there are no changes. So clock genes are flat, similar to what we previously found doing immunostaining for PER2. And again, confirming the hypothesis that affecting the clock function within PHB neurons has drastic impact on the neuronal communication of these targets and potentially in the output signals as well. So as you can see highlighted here are the genes that we found significantly upregulated at CT2 or CT14, and none of them showed changes under these irregular light conditions. I mentioned before GABA cells as the main mechanism controlling the flow of information within the PHB. So we wanted to run experiments where we were able to manipulate exclusively the GABA cells within the PHB. And for that, we use again a similar strategy. Adeno-associated virus, CRE-dependent viruses expressing the DREAD receptor, the excitatory subunit, and TD tomatoes just to identify the cells. And in this case, we use GAD2 CRE mouse lines. Basically, all GABAergic cells in the brain are expressing the CRE recombinase, but we were able to bilaterally inject the virus within the PHB, and therefore only GABA cells in the PHB are now expressing the DREAD receptor. As I explained before, we housed these animals under regular LiDAR cycle for 2 weeks, and we then give CNO at night. Basically, we put the CNO in the drinking water, but they are drinking most of the water at night, so it causes an abnormal activation of the PHB exclusively at night. These 4 weeks, just to avoid any confusion, indicates the time that we waited between the injection and the start of the CNO treatment. As controls, we housed animals under LD cycle, and we then measured the behavior as we did before. And what we noticed is that the DREAD activation was sufficient to affect the behavior of the animals as shown with the tail suspension and novel suppressed feeding test. I included these 2 tests. These are different to what I showed you before. It's just to give you an idea of the battery of tests that we are running, okay? In most cases, when we talked about mood deficits, it's not easy to make strong conclusions with 1 test. So the strategy is to always run a battery of behavioral tests, the more the better, and ideally have 3 or 4 different tests that can provide information about the responses. And we are trying to see responses that are different from control mice. Okay, that's the first step. Then, combining all the information, we can start speculating about which aspects of mood-related behaviors are affected. Again, as confirmed here, the tail suspension test was significantly affected with increasing mobility time, and the latency to eat in the novelty suppressed feeding test was significantly higher. In both cases, indicating mood deficits just by manipulating GABA cells in the PHB. So in this case, there are no alterations of limbic centers, no alteration of the prefrontal cortex of any of the targets that I described before. So the conclusions are highlighted here, where we propose that the PHB can act as a peripheral clock that is directly connected with the retina. So we have a scenario where regular LiDAR cycles controlled the function of GABAergic cells in the PHB that modulate the function of relay cells that eventually communicate this information to their targets, in particular the prefrontal cortex. I put here some rhythms we don't have a precise analysis of the circadian rhythmicity. We are currently working on that. We have 2 time points, so at least we can talk about phasic changes in these centers. On the other hand, under irregular light conditions, we know that the environment becomes abnormal. So there is no rhythmicity anymore, it's unpredictable. And these signals distort the local communication between GABA cells and relay cells, excitatory neurons within the PHP. So we expect that these signals are then routed to output signals, output targets of the PHP, including prefrontal cortex. And this is how we can drastically affect the behavior of these animals. So understanding this local circuitry, I think, would be ideal and essential for designing better tools to manipulate the centers and have pharmacological agents that can directly affect these synchronized circuits, restoring the communication of local circuits that later on impact thalamic centers. So just to finish with this presentation, I wanted to put here some data that we're currently investigating and expanding the analysis. When we did tracing of the PHP neurons, as I mentioned, we confirmed that these cells have local connections that modulate the function of excitatory neurons within the PHP. So this is a virus, again, a Cre recombinase expression in GAT2 cells, and a Cre-dependent virus expressing TDT. So this is the tomato that allows us to see all the axonal and dendritic projections, and synaptophysin is fused to GFP, so we can, in green, identify the puncta that represents synaptic terminals. There is dense innervation and connections within the PHB, confirming these local connections. We have electrophysiology supporting this. But in addition, as you can see in this coronal view, we notice that at least a cluster of these GABA cells have long-range projections that densely innervate areas of the thalamic reticular nucleus, zona incerta, and even contralateral projections to the PHP. With tracing analysis, we confirm that these are GABAergic neurons. So there is a bidirectional communication between GABA cells in the PHB, part of the TRN, zona incerta, and the contralateral PHB, forming a very interesting network of inhibitory cells that can communicate and can integrate, again, information from the internal state, internal cortical signals, together with external information from the environment. If this is the case, we propose that the PHB is the first step in codifying this external information from the environment and therefore communicating these signals to key centers in thalamocortical communication, such as the TRN and zona incerta. So in summary, this is the working hypothesis that we are currently investigating. I started by describing clock genes, central clock and peripheral clocks in the brain and the body. We have this very nice schematic. It's very simple but effective, where the hypothalamus has the central pacemaker within the SCN, and through neuronal and humoral signals coordinate the rhythmic activity of peripheral clocks. But now we have a good understanding of one of these peripheral clocks, that is the PHB, that is directly innervated by the IPRGCs, by the retina. So we still don't know if the SCN is connecting or communicating with the PHB, but we expect that this rhythmic expression of PER2 is in some way modulated by signals from the central pacemaker. If so, these neurons have mechanisms that can integrate these homeostatic signals from the SCN, the circadian cues, together with external signals from the environment through the IPRGCs. In addition, we have a good understanding of the anatomy, the anatomical projections, and some preliminary idea of the function of these cells. I think that this is a model that provides the morphological substrate and the starting point to better understand how light through projections to the PHB can directly affect limbic centers, to affect mood and different complex behavior, but also affecting the function of the thalamic reticular nucleus, zona incerta, 2 key areas that control the communication and the flow of information between the thalamus and the cortex. With that, I would like to thank the people in the lab. As I mentioned at the very beginning, we opened the lab 1 year ago. It's been fantastic time. I was very lucky to have the support of Samer Hattar at NIMH and Richard Lang here at Cincinnati Children's that were essential for this soft and smooth transition. We have a really group of people doing amazing science. We have multiple positions. We are currently looking for a postdoc, so please reach out to us if you have any questions about the position or any question about the talk. I finally want to thank Cincinnati Children's and the funding agencies for supporting this research and allowing us to continue this work. Thank you very much again for the invitation. It was a pleasure to be part of the seminar, and thank you all for your attention.
Great, thank you very much, Diego. Now let's move on to some questions, and we already have two in the chat, but please also think about your own if you want and just put it in the chat. So let me start with the first one by Payal Sandani, who wants to know, or who says, have you observed any changes in neurosensory, retinal, or choroidal thickness between the PHB treated and control samples? If so, it'd be interesting to translate it for the human studies.
Can you repeat the first part of the question?
Have you observed any changes in neurosensory, retinal, or choroidal thickness between the PHB treated and control samples?
That's a great question. I don't have an answer for that. Honestly, the assumption that we're making here is that the retina detects light and this information is routed to the brain. Whether there are local deficits, it's a great question. Yes.
Okay, thank you. Then let's move on to David Sliney's question. I would presume that your studies in the brain can, sorry, if you can also go to the chat and read it there.
Oh, yes.
This is a longer one. I'm still reading out, but it's a bit longer. I would presume that your studies in the brain can model to some degree those in the human brain, despite the fact that mice are nocturnal, have no fovea, and the few cones present in the mouse retina are spread around to be similar to the human peripheral retina. But then the mouse is a dichromat with a UV cone and a green cone and no long wavelength cone. So do you see limitations of your studies to extrapolate to the human?
Absolutely, yes. I'm not trying to make any direct extrapolations. I feel like the comment is absolutely correct. These animals are nocturnal, the retinas are completely different. What we know is that at some point, the retina is processing this information, routing this information to the brain. And these are pure electrical signals, okay? In this case, it doesn't matter if you are detecting blue light, red light, it's a neuron communicating with another neuron. So the research that we are doing, that we are currently doing, is trying to understand this communication process. What is the language that these neurons are using to communicate internal signals and external signals? Animals are nocturnal, absolutely, but at the same time, the T7 cycle is causing a circadian alteration where the animals cannot anticipate the onset of the light phase. And this is similar to what the person is experiencing under a shift work schedule. You know, each week they have a complete dissociation of LiDAR cycles from feeding patterns or social cues. So these aspects are similar. Information with temporal code that now becomes abnormal. And these are the key questions that we are trying to understand. Extrapolations to human research or human pathologies is definitely not what we are trying to do. This is why I mentioned when I was talking about mood deficits that we need to be careful about extrapolating these conclusions. And my only comment and suggestion is run all the tests that you can, because this is very, very informative. And we are trying to see what the normal animal is doing, and now what the animal is doing after the chronic exposure to irregular light. And from there, obviously, we can speculate and try to make conclusions, but we are focusing on this particular data. Yeah, thank you for the comment.
Okay, thank you. If anyone else has a question, just put them in the chat. Meanwhile, I might ask one. So actually, you just alluded to it a little bit with the mood results and like do all the tests you can. So I wondered whether you also I mean, you've just opened your lab, right? So obviously this early work as well. But do you also consider doing some facial expression tests for the like emotions or mood from your animals? Because I've just recently saw this at a different conference, and I thought that was very interesting. So, like, what other tests are you thinking of to test?
Yes. So yes, this is it's an area of research that I really like. I think that it's a lot of debate going on. People complaining about some of the tests. We still don't have a clear idea of what are the proper tests. My opinion, and it's just a personal opinion, it's we need to work on designing tests that can give us longitudinal data. The tests that I presented today are all acute measurements. So we expose animals to 2 different bottles. Do you want sugar or water? It's a couple of hours. The tail suspension is 10 minutes. So that's, to me, the key problem, that we're making strong conclusions based on acute responses. So in the lab, we're working on designing tools that can measure mouse activity and mouse responses across 24 hours, similar to what we do with the wheels. Okay, so for understanding locomotor activity, we need at least 24 hours or even a couple of days. If we can do the same with learning responses as the water maze, but without altering the environment of the animals, that would be ideal. And the same with mood responses. If we can provide them with stimuli that they like or they don't like across multiple days, and we can see the responses, that would be extremely informative. And I think that now with the technology to process this huge amount of data sets, it's something that we can do. We're currently working on this. I don't have any updates to share at this point, but I know that this is where most people are trying to go. So I see really open area for research with many options to explore.
Yeah, interesting. Thank you. Then maybe just one last question. Also, I'm going to ask that. So I assume you basically mainly look at male mice. But do you know, or do you have any hypothesis if that would differ, your results would differ if you'd look at female mice as well?
Yes. Right now, all the analysis we are doing are involving male and females. Honestly, we started using males. Simple reason was doing a lot of anatomical studies was in some way easier to make it more consistent. We are doing very precise injections. So having animals the same weight, same age is essential. But this is not obviously an excuse. We are currently expanding the analysis to females. The preliminary data we have is showing differences between them. So I don't have, again, nothing very clear to share today, but it's something that we need to expand. Going through this data that is already published, I would love to go back in time and do the analysis with females. We know that the circadian responses are different. Housing animals under irregular light, the T7 or light at night, shift work models, there are multiple papers showing that females and males have different circadian responses. So it's possible and it's tempting to speculate that their mood and cognitive functions are going to be also different.
Okay, great. Thank you. Thank you so much. And it's interesting that you're doing that as well, because often, of course, I appreciate the complexity with the cycle and everything. So of course, it's more complicated. Okay, so I think we're at the end. So again, thank you so much, Diego, for this very nice talk. And thank you all for being here today. And I hope we'll see each other after the summer break again, starting in October. So with this, thank you so much and have a nice morning or evening or afternoon, wherever you are.
Great.
Thank you.
Thank you. Goodbye, everyone.